The last talk of today will be presented by Dr. Carsen Stringer. Carsen did her PhD work at the University College of London on computational neuroscience and her postdoc at Janelia. Now she's a group leader at HHMI Janelia Research Campus, where she develops tools for cellular segmentation, understanding high-dimensional visual computations and neural representations of behavior. Today she will present Cellpose and SWIP2P, which are two anatomical and functional complementary algorithms for cellular segmentation. And we are delighted to have you here today, Carsen, and now the stage is yours. Thank you.
Thanks so much. Thanks so much for the invitation. And so I'm going to talk about two algorithms that we've created, Cellpose and SWIP2P, for anatomical and functional cellular segmentation. So what is cellular segmentation? I think you've had quite the intro to it already before my talk. You have an image and you're going to want to assign each of the pixels in cells to two different cells. You kind of want to draw an outline on each of these cells. In the case of a lot of these algorithms, you output this image where there's a different number for the pixels in each cell. For people who might be working with this kind of data, that's usually what's saved as the output. So then you can work with it in another format. Okay, and so why do you need to do segmentation? There's lots of problems in biology that require segmentation: cell counting, monitoring cell type changes, quantifying gene expression, recording neural activity. And so basically all of these different problems have cells that have very diverse shapes. And so we created Cellpose as an algorithm to be able to segment all of this data with basically a single algorithm. So a user doesn't have to train a new network every time they get new data. And then additionally, in the case of functional data, which is neural activity, so this is now data that's X by Y by time. There's a third dimension here. You can see these pixels I've circled here, they have this time course. So you can segment this with Cellpose, and it's actually an option now in our functional segmentation pipeline, but it actually helps to use temporal information. Like Andrea talked about too, if you have two overlapping cells, you can distinguish them from each other based on differential activity. So we have another algorithm to do that called SWIP2P. And so how are we going to solve this general segmentation problem? Well, so deep neural networks, as have been discussed, can perform segmentation. And the big commercial use of that is for self-driving cars. They make networks like MassCarCNN are designed to both do instant segmentation, find all the cars in this image, and then also semantic segmentation, which means label what type of object is in the image. And so these algorithms are often trained on a database called COCO. And so this is a really large database of images that these algorithms can be trained on, but there isn't actually a COCO for cells. There is a great database for nuclei that you can check out from Ann Carpenter's lab, but there isn't this sort of COCO for cells. Additionally, MassCarCNN has these two different sort of steps that they put together to do this segmentation, which maybe is not the most optimal for this cellular segmentation problem, which is really an instant segmentation problem and not necessarily, in our case, we don't necessarily care about solving the semantic segmentation problem. And so we're basically going to try to create a large database and also create an appropriate loss function for cellular segmentation. And so to get a very large database, we basically searched Google for lots of images of cells, just literally just searching for cell. And Mihaly Mihailos in the lab labeled all of these images in our GUI, which allows you to kind of circle cells quickly. And so we have over 70,000 cells in the database, which you can download on our website. The link is in our paper. And so now we need a neural network that's optimal for cellular segmentation. And so basically you have an image that looks like this of cells, and we're going to turn this into each cell into its own little dynamical system. And so this flow legend is the sort of legend that people use in the optic flow literature for people who are familiar with it. So each of this HSV color map is corresponds to each of these different angles. And you can see these cells are now basically, there's flows in each of these cells which point towards the centers of these cells. So you can see these little dynamical systems. So if I was to start a dynamical system where every pixel is a little particle in the system, and I started running the dynamics, they would all converge to the centers of their cells. And so we're going to train a deep neural network to learn this kind of proxy representation for these cells, these flows. And so this is what this image looks like. And we think this is a useful representation for another reason, because it helps with avoiding merges of cells. So if you look at the boundary between these two cells, you can see the flows are in complete opposite directions. So we're really forcing the network to learn that cells are distinct, and we think that's kind of an advantage that this representation has over using something like a UNet or something like that. And then it also gives us access to learn less convex shapes. So the flows can kind of flow from the end of the cell to the center, so they can flow around corners a little bit. And so you can have complex cellular shapes that can be learned here. So here's an example of what I was describing before, where every pixel is run as a particle in this dynamical system, and how they all converge to points which are at the centers of the cells. And we also have a third, so Cellpose predicts two maps for these flows, the X flows and the Y flows. I'm just showing them here in this nice picture. And then it predicts a third thing, which is cell probability, which is commonly output by UNet architectures, which people might know. And that kind of that's filtering this image here. So only the particles above a certain cell probability, the pixels above a certain cell probability, are used in the dynamical system. And then finally, that outputs an image where every cell has its own label. Okay. Okay, I think this question is, okay, yeah, sorry, that's still a question from the last talk. Okay, so now we're going to try a proof of concept with this new type of network loss function. And we're going to do it with a specific set of cells, this cell image library database, where there's 100 of these images with many cells. And so we trained Cellpose, Stardust, and maskRCNN to segment these cells. And so the ground truth is shown as yellow and the prediction is in red. And so these arrows here are kind of pointing out errors that these other algorithms make that Cellpose doesn't make on this image. So for instance, Stardust misses this cell, perhaps doesn't follow all of these long dendritic structures in these neurons. maskRCNN may oversegment things. And so then we can say, okay, qualitatively, it looks like Cellpose did a better job, but quantitatively, how do we benchmark these kinds of algorithms? People often use an accuracy measurement, which is the number of true positives divided by the number of true positives plus false negatives. So how do you define a true positive? Actually, that kind of depends on how accurate you care about being. So people often use this intersection over union criterion. So you say how much area overlaps between maybe my ground truth mask versus my predicted mask divided by the total number of pixels that are surrounded by these two masks. And so for this cell, this is a very high intersection over union. And then for some of these, it'll be closer to like 0.5. And so you can define a true positive as, well, I got most of the cell, that's all I care about. If I'm doing cell counting, that's mainly what I would care about. But if I care about getting precise boundaries, I might care about getting accuracies that work well, where I care about true positives only at a very high intersection over union threshold. And so we make these curves, which are also common in the field, where we compute accuracy as a function of this threshold I was describing. And so accuracy goes down as we make it harder and harder to define a true positive. But what you can see is Cellpose in the green outperforms Stardust and maskRCNN across these IOU thresholds. And so you have higher accuracy cellular segmentation at higher IOU thresholds, signifying we got these boundaries well, and also at the lower IOU thresholds, signifying we were able to detect we were not over merging or over splitting cells. Okay, and so now the proof, the ultimate thing we wanted to do was create a model that worked for lots of different data. And so now we're going to use all of our data sets, not just the cell image library. And so we've trained our data, we've trained our model on all of these different images, and we're going to look at the performance. And again, what we found was Cellpose outperformed Stardust and Mascara CNN. You can kind of see this is an example of an area. There are 3 cells here that Stardust merged and Mascara CNN also merged. And so again, I can show the quantitative proof that Cellpose outperforms these other algorithms on this type of data. And then what we also care about is that we've trained on all of this data. Did we lose performance on some of these other images, like the cell image library? And what we found was that no, actually, if I overlay the curve from the last slide, you can see that we didn't lose performance on the specialized data. So the network had the capacity to be able to segment a wide variety of images, basically. And so you can use this algorithm on lots of different types of images. I'm just showing the performance on these like cell-like images, but we also have rocks and stuff in our data set that you can also segment. And you'll see a lot of those here. You can see the garlic cloves here. So these are all examples of test images that were not in our training set that Cellpose did a good job of segmenting. Some sea urchins, things like that. And also cells, which are the more important thing. Okay, and so then the last part of Cellpose is exactly what Daniel was alluding to in his talk, was that getting 3D data is very difficult. And so we've labeled all of this 2D data. And so now we want to try to use our 2D model, our 2D training data to inform a 3D model, rather than having to collect 3D data where I have to circle 10 times as many, basically, ROIs to get a single cell segmentation. And so we're going to do this, we're going to take our volume and segment in 2D, each of our slices in 3 different ways. So we have our volume in X, Y, and Z. We're going to slice in X, Y, and take each of these Z slices and run Cellpose on them. That will take basically XZ slices, and we'll also take YZ slices. And so then we run Cellpose on each of these slices, and they're going to give us flows. So the XY slices give us flows in X and Y. The XZ slices give us flows in X and Z. And the YZ slices give us flows in Y and Z. And so now we can average flows across these different segmentations from these different angles. And so this is going to allow us, so you can imagine if 2 cells are kind of on top of each other and it's hard to tell if they're the same cell or not in one plane in XY, that might be distinguished in XZ actually. And so the X flow from the XZ plane averaged with the X flow from the XY plane might be more accurate. Basically, we're hoping that this 3D information gives us better context for the cell shape. And so when we average, basically average, for instance, XY, for the X flows, we average the X flows from the XY and the X flows from the XZ, for instance, and so on. Then we get a picture that looks like this, where we have the X flows and the Y flows from these averaged slices, and then we have the Z flows averaged from these 2 slices here. And so the Z flow, the color scheme is a little weird, but so basically, like, red means you're below a cell, the cell center, and so you got to move up, and then blue would mean like you're above the cell center, and the Z flow is down. And so we can use these 3, now we can run our dynamical system in 3D, not just in 2D, and see where these pixels ultimately converge to, and segment using those convergence points in 3D. And that gives us a picture that looks like this. And you can make it rotate in image J if you want to. I think a nicer way to look at it, which is a way you can scroll in lots of, in our GUI, in the Napari GUI, other GUIs like that, is to scroll in Z and look at the masks on top and check that everything looks like it's segmented correctly. You can see how it's able to segment these cells that are right next to each other. And this is an example of tissue for that's being used for single cell RNA sequencing experiments. So this kind of accuracy is really useful for these sorts of experiments to make sure that you're assigning RNA expression to the right cell. Okay, so how well did we do? So this version where we've trained Cellpose on 2D data and then run these flows in 3D, that's called Cellpose 3D. That's this green curve. We outperform examples of algorithms that have been trained in 2D as well, but don't use 3D information. So for instance, these stitching models. So we've trained Cellpose in 2D, and then we run it in X, Y, and then just stitch the masks together. That doesn't do quite as well as using 3D information. You can do the same thing with MassCar, CNN, and Stardust. Those don't perform as well either as the full Cellpose 3D. And then there's 3D versions. We trained a 3D elastic model on a held out portion of the volume, and that also didn't do as well as Cellpose on this data. All right. And so finally, I'm just going to show you how our GUI works. And feel free to put in any questions now. If you have any questions about Cellpose, I can pause for a second and then we can move on and I'll talk a little bit about Sweet2P. So here's an example of running an image in the Cellpose GUI. You can drag and drop it, run the segmentation. You can modify the segmentation. So this user, me, is going to delete a cell and then draw some new cells. And you can also, we have this button called send manual segmentation to server. So people can send their segmentations to the server to make our model better. And actually lots of people have done that. And so we have actually released a new model called Cyto2 that you'll see in the GUI that uses these new images. And you can also run it in the command line interface with Cellpose as well. And so that might help. A lot of the images that people sent were maybe a little more low SNR than what we had in our original database, so it might help with that. It didn't end up actually helping with our original test set in terms of performance, adding these new images. Okay. And then I think, okay, I'll answer these two questions in a second. Okay, so after I'll just conclude. So Cellpose has out-of-the-box performance for many types of images. It's easy for us to integrate labeled images from users. So if you use our native GUI, we can get your segmentations from you, basically inside the GUI. We also have integration with other platforms like ImagePy, Enjoy, Napari, and Cell Profiler. And so, yeah, and so big thanks to Marius Pakisariou, my main collaborator on this project, and also to Tim Wang and Mihalos. And yeah, I'll take questions now, and I guess they'll just be in the Q&A box.
Yes, actually, we have two questions. Don't know if you want to read them.
Sure.
Okay, great.
So Gebhard asks, is Cellpose available for MATLAB? No, but I think you can run some Python things in MATLAB, but hopefully the GUI is using our GUI or another GUI to run Cellpose is intuitive enough that it's doable without using MATLAB. And then also the outputs, you can save the outputs of Cellpose in various ways. You can save text outlines like for ImageJ, you can save masks as PNGs or as TIFFs. And so then you can load those into MATLAB and use them for post-processing. And then there's another question from Federico. How does Cellpose deal with cell overlap? So that's a good question. So we don't deal with overlap, basically. And so every pixel can only be assigned to one cell. And so if you have an image with a lot of overlap, then the best algorithm right now, kind of the state of the art, is to use an algorithm like MassCar CNN. But in practice, I think, well, depending on the resolution of your imaging, like particularly your kind of Z resolution, hopefully you can distinguish pixels between two cells. Or for instance, if you don't have a great point spread function in Z, maybe you can take a Z stack and be able to distinguish exactly where those cells are. Because a lot of the overlap, I mean, two cells can't actually physically be over, like they have their own space. So if you have 3D information, maybe you can distinguish exactly where those pixels are from those two cells. Yes, Federico says yes. Okay, cool. And then for the Cellpose with Sweet2P, I'll show that next, actually. So are there any other questions about Cellpose? So that's a good question.
I think not. So I think we can continue, and then maybe at the very end we will try to answer also the other possible questions on Cellpose.
Cool. Okay. Okay. So there's still so we've shown an algorithm that's very useful for cellular segmentation, but it might not necessarily be enough, particularly in the case of calcium imaging. You have a lot of cells that maybe you can't actually see in an anatomical image. So here's an image of an like the average image across a recording of calcium imaging, and you can see kind of we've labeled the cells with these dots. Those are like the centers. This was before Cellpose. I just clicked on where I thought the cells were, basically. And there will be cells that you can't actually see in this mean image that get detected with a functional algorithm, which can actually see which pixels are active at certain times and are active together. And so this is just kind of an example of like here's a couple of cells that were difficult to see in the anatomical image, but have clear traces. So the blue is the activity of this neuron, and then the gray is the neuropil, which is the average of the activity surrounding that ROI. So hopefully this activity is higher than the surrounding background activity is the goal. And so when these traces look pretty good, that means like higher than the baseline, then it means you've done a good job of detecting a cell, for people who aren't so familiar with this. But then with the anatomical algorithm, so these are two cells I missed by labeling by I, and then there's some cells that I labeled that maybe don't necessarily have great activity, that kind of aren't active during the recording, that maybe I didn't care about detecting. And so we want an algorithm that can take into account this functional information. And so we created SweetUP, which takes as input a lot of different formats. And the first thing it does is run motion correction. So you have this image here on the left is the unmotion corrected field of view. And so a lot of these recordings are done in vivo. This is in a mouse that's awake. It's free to run. It's often running. And so you might have some of these movements that you need to correct for. And so the frame on the right is the corrected one. And if you don't correct them well, then you might think that movement is actually activity, which is bad. And then the next step is the segmentation algorithm. And so there's also an option to run Cellpose and SweetUP, which I'll show in a second, but we also have our own functional algorithm, which finds, in the case of this recording that I'm showing, it finds twice as many cells, the functional algorithm over SweetUP, or sorry, over Cellpose. And then we extract the signals from these cells, which are basically just summing the pixels in each of these masks. And then you can save the output in a variety of formats. And then finally, we have a GUI to look at the data. And we have in the SweetUP library on GitHub, you can see we have a new Google Colab notebook that allows you to run SweetUP in Google Colab if you choose to. And so that might be an easy way to test out how well SweetUP works on your data, and it runs quite fast. So here's an example of 4,500 frame recording where it runs in under 10 minutes, basically, to process all of this data. Okay, so for cell detection, you can use something like Cellpose, and so the default in SweetUP is to use the max projection image, which is the max. Each pixel is assigned the maximum value of the pixel over time. We do something a little more complicated than just using the max projection image, but it's basically using this image to do the segmentation. And this generally will work the best because it actually does combine a little bit of that functional information because it's taking the maximum of the pixel over time. So it's not just the mean image. And so if you do something like that, here's an example on the left of hippocampal data. Cellpose does a good job on this. And actually, we do have some hippocampal data in Cellpose, so that might be part of the reason why it does fairly well with it. Not from this, actually from a completely different lab, but yeah, so this is data from a different lab that's been segmented from the training data from Cellpose. And then here's an example where Cellpose might work well in really deep imaging. where the signal's kind of low. In these low SNR cases, it might be helpful to use Cellpose because the masks might be kind of fuzzy. It might be difficult for the algorithm if there's very few active frames for it to get a good sense of the shape of the mask. And so that might be another case where Cellpose might work well for you. And so here's an example. This is, the field is called Anatomical. If you're using the Sweet2P GUI, that's where you would put in this option for Cellpose to turn it on. But keep in mind, you need to install Cellpose for it to work in the GUI. So you should install Cellpose first and check it out, and then try it in Sweet2P. Okay. So then how do we use this functional information to do a bit better than just using the anatomical information? So the first thing we do is we take this movie, which is X, Y, T, because we have this time dimension, and we're going to bin it in time with the bin size of the GCamp timescale. So basically we're just trying to reduce the size of the data at this point, so to make it manageable, and also because there's kind of degenerate information in this volume, like so there's a correlation in time of these pixels that's proportional to the GCamp timescale, because the cell will turn on and then it will shut off slowly. So we can kind of assume that if we bin in time, we can kind of maybe like smooth a little bit, make it a little higher SNR, and also reduce the size of the data without losing information. So if you are, this is one point, you can bin more in time by like artificially changing. For instance, you can change the your sampling rate or your tau in Sweet2P, and that will actually change how much it bins in the algorithm. So if you're having trouble finding cells, that's a trick to kind of give the algorithm higher SNR data, which might help. Okay, so once you've done that, we high pass filter in space and time to try to really focus on events that are localized in space, ignoring the low frequency neuropil, the low spatial frequency neuropil, and ignoring long timescale processes, basically looking for peaks in calcium that are fast. And then what we do is we bin these maps in space. And this is just a fast, we do it this way to make it fast and more efficient, but we're basically running a uniform filter in space. So we're like, if you have this like 3x3 filter that you're summing all the pixels in that 3x3 area, and that's your new little movie here. And then you sum again in 3x3. And basically what's going to happen as you sum in a 3x3, if those pixels are correlated, that 3x3 pixel sum is going to be bigger than it was on this bigger map here. And so the peaks are going to come out at the size at which the cell is at. So like you can keep binning, and if it starts binning noise around the cell, then again that peak is going to go down. So ideally, when you do this kind of binning process and you take the max over time, this is what these little maps look like. The maximum will be found kind of at the size of that peak of that cell. Kind of, if that cell is a 10 by 10, then it'll be biggest on the map that's, we have a map that's like 12 by 12 summed, if that kind of makes sense. So then we start hunting for these peaks. So we try to find the biggest one first, and depending on what map it's found on, we initialize the cell with the size proportional to how big that map is. So if it's on one of these maps that's super downsampled, that means it's a big cell because it had its biggest peak when we summed a ton of pixels. And so, oh yeah, here's just a representation over all the maps, just kind of trying to combine them together and smooth them. But that's not actually used in the algorithm. That's just something you see in the GUI. But yeah, so we initialize the cell with this square around the peak that's proportional to which map it's found on. And then we don't use all of the time points to grow kind of to make the shape of the cell, because we want to be able to find, for instance, if we want to find overlapping things or things with different time courses, then we only want to focus on the time points when this specific seed that we found is high. And so we just focus on these active frames here above a certain threshold, which is proportional to kind of the peak on the map. And we just focus on these active frames and use those active frames to grow the cell. And so basically we take these frames, we average their activity, And we basically take all the pixels above, in this case, it was kind of arbitrary. We take like above the max divided by 5, and those are the pixels we include in the cell. And then so we start changing its shape, and then we accordingly can change which frames we're focusing on as we change the shape and grow the cell. In this case, this is not a very exciting cell shape, but it can learn things that are a little more interesting looking as it grows iteratively. And so it can learn these little dendritic things. And yeah, I think I'm sorry if I'm going a little bit over time. I'm almost done. So basically then in the end, you have this map, which is where now, so each of these little cells we found is colored, the pixels are colored by the cell that it's in. And oh, I don't have the image here, but this is 2 times as many cells as we found with Cellpose. So this is now 1,400 ROIs compared to only around 700 ROIs. And by regions of interest, I mean cells. We can do this on large scale imaging data. You can find thousands of cells simultaneously. So, and the nice thing about SweetUP is that it's fast, and there's an option that we use to run it even faster, where every plane gets sent to its own set of cluster cores. And so basically one of these recordings with 50,000 neurons can run in like 15 minutes. And so that means, again, like this analysis loop can be very fast with the data. Not quite as fast as processing online, which is pretty exciting, but still fast. Okay, and then finally we can grow. This growing feature of SweetUP can be really useful for finding these interesting dendritic shapes. So for instance, these are Purkinje cell dendrites in cerebellum that SweetUP was able to find and trace correctly. Okay, and then we also have a GUI, which is also built in PyQt and PyQtGraph, like the Cellpose GUI that you can use to analyze your data. And it's really useful, even if you run SweetUP in something like Google Colab, or you can download it and look at it here, because you might want to look at, for instance, how well did the registration do? We have options where you can view the movie of the registered data versus the unregistered. You can check that the motion correction is good, and all these different things that are really useful for you to benchmark if your algorithm is working correctly. And so basically, I didn't go through our registration benchmarks, but our registration is very accurate. We can find lots of cells with the cell detection. In certain cases, Cellpose might be helpful. I didn't go through this, but we also have a new PCA denoising option. And we have a beta tested classifier, a ground truth classifier, which I also didn't show in the short talk. And then kind of what's next in SweetUP, again, things I didn't really talk about, but if you're interested in adding to the code, we're happy to take pull requests if you're interested in adding different aspects to SweetUP. And so again, big thanks to my main collaborator on this, Marius Pakisario, and also last summer we had some software engineers who worked with us, Nick Del Grosso and Chris Key, so thanks to them too. And thanks everyone for your time and attention.
Thanks a lot, Carsen. Amazing talk. And I'm very happy to see that now we can use Cellpose combined with C2P. Very happy about that. Let's see if there are some questions. Probably not so far, so I maybe can start. I was wondering if you ever used maybe C2P to analyze calcium imaging and to segment different type of cells other than neurons, such as astrocytes, for instance. Have you ever tried to segment this kind of very complicated cells, which have a very complicated shape, let's say?
No, I haven't. I actually haven't seen astrocyte data and segmented it myself. I don't know if other people have used it. But I guess, oh, sorry, I don't think my screen's changing, but I just wanted to say, like, this, it can grow arbitrary shapes, and it's not restricted necessarily in space. And so it can create ROIs that maybe would be useful for astrocytes. If the processes are really long, Cellpose will struggle with them. So that's why it might actually make more sense to use SweetUP.
Okay, great. And oh, we have Andrea Giovannucci with his hand right? Do you want to say something?
Yep. Thank you, Carsen. Cellpose is amazing. We use it a lot. I have a question. Well, if since you use it in, you show that it can work in 3D, and if you look at the calcium imaging data in some sense as a three-dimensional.
Oh yeah.
Did you try to do that? You know, use Cellpose, but now trying to capture the activity in the third dimension.
No, so we don't take cellular functional stacks in such density that we would use Cellpose in that case.
No, what I mean is not in 3D in that sense, meaning
where time
the
is
third dimension is time. Yeah.
Yeah, we haven't actually tried that, but it's an interesting idea. I think it makes more sense with cells migrating than with these short kind of events that we're trying to explain. I think it doesn't enforce the sparsity that we expect with the traces. Like you have your sparsity penalty, then I think enforcing those is really useful. And SWE2P does that by focusing just on those active frames. That's how we enforce our sparsity. So I don't know if it's the right approach for that, but potentially for cells moving or migrating, it might be.
Thanks.
Yeah, good question.
Okay, so maybe perhaps I have another question. So suppose we have double staining or in principle multiple staining. Is there any attempt to build a consensus between the two different segmentation we might end up using Cellpose, either Cellpose or SWE2P?
To combine them? Yeah, I think that's something you would have to do post hoc, and it's not currently an option in SWE2P to load in your own masks. Like that's something that yeah, we'd have to happy to take like a contribution on that or request on that. But yeah, that would be something you would do offline, because there's a lot of there's a lot of qualitative judgments you're going to make on that, basically, in terms of which cells you accept or don't accept. But generally speaking, so I've kind of tried like using Cellpose to seed SWE2P and vice versa. But if your data is really high SNR, I think in the end, using the functional algorithm is the best way to go, kind of no matter what. I didn't really see big advantages of adding Cellpose on top of SWE2P with kind of the high SNR visual cortex data we have.
Okay, thank you. I think we have another question from Marco Salamanca. I don't know if you can read it.
Yeah. So Marco says, how does SWE2P perform with different neuropil and signal to noise ratio contamination levels? Yeah, so the first thing we do is we high pass filter in space. So getting rid of kind of the neuropil contribution to these signals. But if the SNR is really low, it will struggle, because it basically, when you, hold on, let me go. Kind of like I was describing, where we create these ROIs, we're only averaging the time points when the cell is active. And so if it's only active on a few frames, if you have a really short recording and it's also very low SNR, then it will struggle to make these masks, and these masks will look kind of like fluttery and flaky looking. And so, because we don't have any smoothing on the masks post hoc. And so that might be an example where you'd want to use Cellpose for your masks or something else, because we don't have this spatial smoothing of our masks that we enforce, because we prefer to like try to be as close to the data as possible and not put any kind of constraints on what we're looking for or anything like that.
All right. I think we don't have any other questions. So we can move on, and so Anna can conclude the meeting. Thanks again to all the speakers.
In the meantime, if anyone has other questions, you can write, or you can also write to us later on, and we can try to address in our website your question. And I would like to thank once again the speaker, also to remind everyone that they and their group are developing an open source tool that is something really, really important, fundamental for the scientific community, and not taken for granted. And also I know that today the participants are coming from different countries, And if any one of you want to say in the chat from where it's coming and taking part of this, also would be nice. And I think that's it. And now the organizer and the speaker will meet in a separate virtual room to have a kind of very informal round table. I don't know if anyone in the audience really would like to take part of this round table. Please raise your hand. Maybe we'll try to include you. Let's wait a few, 1 minute. Otherwise, I can. Okay. Okay. Thanks then, everyone. We close this webinar and see you next time. Thanks. Bye.
Thank you.
Bye-bye.
Bye.
Good.